future doctors, welcome back to Best Medicine's GCSE AQA Biology series, in which we're going to be talking about preventing and treating diseases. In the previous chapters, we already looked at how we can prevent diseases by reducing the risk factors, like having a better diet, having a sedentary lifestyle, eliminating smoking, eliminating alcohol use from our lifestyle. Another thing that we've looked at for prevention is decreasing the rate of transmission of pathogens, right? So when we are able to, for example, maintain a good hygiene or eliminate vectors, all of those would basically help in reducing the spread of pathogens. So that's the kind of prevention that we already covered. However, we haven't covered vaccinations in detail, right? The number one thing that comes to mind when we talk about vaccinations through some intrinsic body agents that will improve our immunity, well, the number one thing that comes into mind is vaccinations. So vaccinations, how does a vaccine work? What do you think? We already looked at this, right? When we were doing the white blood cells in the circulatory system chapter, we said that there is B lymphocytes, right? B lymphocytes. And what happens is that B lymphocytes get a first exposure, right, of a particular pathogen. And when they have this exposure, they differentiate into two kinds of cells. They differentiate into plasma cells that make antibodies, and they differentiate into memory cells. Now, memory cells store the information of the antigens that were present on the first exposure's pathogen. And so if the same antigen containing pathogen comes in again, then you're going to have a second exposure of the same pathogen, right? And a second exposure or further exposures, you already have memory cells for those antigens. So if you have those memory cells, the memory cells will very easily and very quickly recognize that, okay, it's the same pathogen because it's got the same antigens. And so this will very quickly differentiate into plasma cells, making antibodies. Okay, so the difference between the first exposure is that this is slow and takes a lot of time, while the second one is very, very fast because you already got the memory cells. The first exposure, it takes time because the B lymphocytes, they have to create antibodies that are complementary to the pathogen, and so it takes some time to be able to get that perfect shape to be able to bind the pathogen. But once it's already trained, once you have the memory cells, then the second exposure would result in a very fast response. So if you get infected a second time, a lot of the times the response is so quick that you don't even develop symptoms at all, right? You don't even develop symptoms at all. You might not even know that you're infected. So that is first exposure, second exposure. Now here's how vaccines come into this. Vaccines, they provide a pseudo first exposure. They provide a fake first exposure, right? So vaccines contain antigens, right? Either they can just contain antigens, or they can contain weakened pathogens, weakened or dead pathogens. That will then eventually cause the first exposure response, which will eventually cause production of memory cells, and then you will have those memory cells after the vaccine. So that is how vaccinations work. They treat you with either the antigens of a known pathogen or weakened dead pathogen itself, so that you're able to make memory cells for it, so that if the real pathogen comes in, you already have memory cells for it, right? Your system is already ready for it, and you get a very fast response. Now, a very good example of this is COVID. With COVID, you normally take 14 days to get better, but if you've already been vaccinated, then you already had the antigen exposure, and so you already have the memory cells for it. So if you already have the memory cells for it, then you're going to have a very fast response, and you might not even show symptoms for a COVID reinfection, okay? So that's how vaccines work. Over here, it's illustrated in text. A weakened or dead pathogen with antigens or antigen itself is injected into a person. So what it does is it kind of provides a pseudo first exposure, a sham of a first exposure. This antigen is recognized by B lymphocytes in a slow first response, which makes plasma cells to make antibodies against it, and memory cells. So memory cells are the key thing here to remember the pathogen for a possible second exposure. So if the real pathogen now comes in, earlier we only had a pseudo first exposure, just the antigens, or a weakened pathogen. If the real strong pathogen infects the vaccinated person, the memory cells are already there, right? So it's a second exposure, and memory cells will quickly differentiate into plasma cells, which quickly makes antibodies to quickly get rid of the pathogen before it even has a chance to cause any symptoms occasionally. Now, examples of common diseases with vaccines include chickenpox, poliovirus, measles. We already had a look at what measles is. Remember, it's a viral illness that causes a rash, fever, right? And it has a vaccine. And COVID-19. Tetanus also has a vaccine. Herd immunity. What is herd immunity that is achieved with vaccination? Herd immunity means having a specific number of individuals to be vaccinated so that the number of susceptible individuals in the population is decreased. So you have basically a large proportion of the population is vaccinated, which means that the susceptible individuals are very less. That is called herd immunity. This will eventually lead to an eradication of the entire disease, because if you're not able to transfer the disease to the next person, then the disease has nowhere to go, and it eventually gets treated in the affected person, and the disease just gets eradicated from that population. So that is herd immunity, right? If you don't have any herd immunity, then you're not able to, then the people that are susceptible are just too much, and everyone can have the disease. But if you do have herd immunity, for example, green ones are vaccinated, then only a few options are left to transmit the disease. And if you're not able to meet those people and transmit the disease, then this person will get better over time and before they even have the chance to spread the disease, which means that the disease will eventually be eradicated. Now, a very popular example of a disease that has been eradicated from the world due to herd immunity throughout the world is smallpox. The smallpox virus has been eradicated by this. So again, herd immunity occurs when a large proportion of the population becomes immune to disease. This provides indirect protection to those who are not immune. Herd immunity works by reducing the number of susceptible individuals in a population, making it harder for disease to spread from person to person. Right, so if a significant proportion of the population is immune, the chances of an infected person coming into contact with a susceptible person decreases, and it slows down the transmission, and eventually the disease as a whole is eradicated. Now, there's also something called ring immunity. Let's have a look at what ring immunity is. Ring immunity basically means that you have a particular area. Let's say this is a city, let's say, right? And then all these are farms, right? Let's say this is or maybe this is like a village or something, and then all of these are farms, right? So these are all farms, the circles, and then each of these farms have cows. And let's say that one of those cows, maybe a cow here, right, gets a disease. So if a cow here gets a disease like a viral illness, here's what you do. Okay, you find out, okay, this cow has a disease. What you do is you cannot just go ahead and start vaccinating the entire village or nearby villages. No. What you can do is that to prevent this from spreading, first of all, you need to contain the cow right over here and not let it go anywhere. And second, what you do is you provide immunization, vaccination to nearby farms only, in a ring. So that you form this ring of protection, this ring of herd immunity, in between that affected farm, for example, right? And that is called ring immunity. And we also call it ring vaccination. Okay, very simple. The other way to obviously prevent disease and treat them is drugs. Drugs, not the ones that you are thinking about, right? Drugs means medicines. So several drugs are common among our society. Examples include NSAIDs. NSAIDs, they help with pain and fever. For example, aspirin and Tylenol, which is paracetamol, and ibuprofen. And antacids, for example, omeprazole, are also very common among us. Now, antibiotics are medications that treat bacterial infections, right, by either killing the bacteria, they kill the bacteria, or stop their growth. So those are bacteriostatic antibiotics. Again, antibiotics can either be bactericidal, which means they kill the bacteria, or bacteriostatic, which means that they are able to stop their growth. Now, examples of an antibiotic would include penicillin. The way they work is usually they target the bacterial components, for example, cell wall. Most importantly, penicillins, for example, target the cell wall and destroy them. Or protein synthesis. A group of antibiotics, I can tell you this, however, you don't need to know, but aminoglycosides, for example, right? Very common. You might have heard of them before, or some examples of them. These are very popular and they stop the protein synthesis. Or disrupting essential processes for bacterial survival without affecting the human cells. So antibiotics cannot treat viral illnesses. That is very important. Sometimes, when you get a viral illness and you go to the doctor, he will give you some antibiotics. And it's not because he's giving it to you to treat the viral illnesses. Instead, what they do is they prevent any bacteria to cause an infection on top of the viral illness. That's why they give you this preventive antibiotic course. Okay. For example, when I was young, right, I had chickenpox, which we've already talked about a little bit. It's a disease that has a vaccination. I didn't have vaccination, so I did get chickenpox in a very severe case of it. So when I went to the doctor, they gave me antibiotics on top of it, so that I don't get a superimposed infection on top of the viral illness that I already have. That was chickenpox. Okay, so there's a rising concern now over antibiotic resistance, something that we already had a look at before as well. Now, antibiotic resistance can result due to, and I already asked you a question on this before, so hopefully you guys have answered that question in the previous video, but the answer to that question is given in this text here. So prolonged or irresponsible use, for example, by not finishing a course, that's one of them, gives opportunity for infectious bacteria to survive and mutate. Either you don't finish a course, right, or you start abusing the antibiotics for even mild illnesses, right, things like that. So the mutations can, by chance, lead to development of resistance against antibiotics. And a very common, very popular example of antibiotic resistance is something called methicillin-resistant Staph aureus. Staphylococcus aureus is a bacteria that is very popular to be resistant against a common penicillin, methicillin. We've already talked about this once as well, and so here's a repetition of it, and it's also for short-term, for short form, it's also called MRSA. MRSA is a very popular bacteria that has developed resistance against methicillin, right? And so to treat this nowadays, we use something called vancomycin, a big gun antibiotic, right? Vancomycin is more expensive, and nowadays we even have Versa now, vancomycin resistant antibiotic, Staph aureus. So you see, there's so many problems that are arising because of antibiotic resistance, because we are overusing them or we're not finishing their course. So that's very, very important to finish their course and to be able to not abuse them. Okay, so that finishes our section, and here's a meme. Teacher, what is the opposite of antibiotic? Student, unclebiotic. Unclebiotic. Antiviral should be the answer, right? Teacher, student. Run away. Hopefully one of you guys can use this joke in your class. But then be aware of this situation. Anyways, discovering drugs. This is a very, very popular board question. Discovering drugs. Where were some of the drugs discovered from? Okay, very popular question. Digitalis or digoxin is one of the drugs that helps strengthen the heartbeat. Okay, so it's good for heart failure patients. And this was discovered in foxglove plant. Okay, foxglove plant. So they can literally just directly ask you, where was digoxin discovered, or how, or what plant, and you need to know, or you need to write this answer, or they might give you a couple of options as well. So choose foxglove from it. Aspirin is a non-steroidal anti-inflammatory medication. Anti-inflammatory means it decreases inflammation, which means it decreases pain and swelling and fever. And so aspirin being a non-steroidal anti-inflammatory medication or drug, this was discovered in willow bark. Okay, bark of the willow trees. So these two were discovered from plants. Penicillin, very, very important. Penicillin, which is probably the most common or most popular antibiotic. This was discovered when Alexander Fleming accidentally spilled some mold onto a bacterial petri dish that he was growing. And he discovered that the mold killed the bacteria. So this was just a coincidence. He was experimenting and stuff, and then this accident happened, which was a wonderful thing for all medicine, that he spilled some mold and it ended up killing the bacterial colony. So the mold was penicillium, and so he named the antibiotic component of the mold, which would kill the bacteria, penicillin. Okay, comes from the penicillium mold, penicillium fungi. So that is another one of those questions that can come up. How do we develop drugs? How do we develop new drugs? How do we develop drugs to be approved to be used in hospitals? This is a very important and a little bit difficult topic, because in this one you'll have to memorize the phases of trials and researches. Okay, so to develop a new medicine, we need to make sure that it is safe. First of all, it is stable, which means that it can be stored. It is effective, it works. Not only is it safe for the individuals, it means that it doesn't have side effects, right? But also it treats the disease, and it is removable by the body. You don't want the drug to start accumulating in the body, because that would be disastrous. So process in order. Here's the thing. You first discover the drug, right? Drug discovery. This can be taken from many different books or people's normal experiences, etc. For example, the way aspirin was discovered was that if you chewed on willow bark, it would relieve inflammation and pain, right? So they started researching on it and so on. So once you discover the drug, then you start testing if it actually works and what is the component and all those things. So in all testing which we plan for approving the drug, there will be a standard set of individuals that receive the drug to try to test it and receive something called a placebo. And you need to know what a placebo is for the sake of AQA exams. What is a placebo? What is a placebo? A placebo is something like a sugar pill or a fake treatment that doesn't really do anything. Okay? It doesn't really do anything. So, for example, I'm conducting a clinical trial, or you and I are conducting a clinical trial, right? So you and I discovered that we read about a drug, maybe in some Chinese old book, that there's a drug that treats maybe malaria or something, right? And then you and I try to investigate if it actually works or not. Then what we will do is we will plan an investigation in which we get a large set of individuals with malaria and try to see if the drug works on them or not. Now here's the thing, people undergoing this treatment will receive either the drug or no drug, right? Something that looks like a drug but doesn't really do anything. That is called a placebo. So it's given to people in experiments to see if they feel better or feel any side effects, because they believe they're getting the real medicine. Okay, so people don't know that they're getting the placebo. I hope you understand that part. So once we discover the drug, then we have preclinical testing in laboratories. So preclinical testing, you can just remember this, in labs. So this means that you test a drug in labs on cells, tissues, in mice, etc. So for example, if you're trying to test a drug for cancer, you have some mice with cancers, and you give them that drug and see if it works or not. And if it does work, then you jump on to the next phase. Okay, you get approval and then from official authorities, and then you jump on to the next phase. That is called the phase 1 clinical trials. In phase 1, you test it on healthy volunteers only. Okay, you're not testing the drug for efficacy. You're not testing to see if it works. In phase 1, you're only conducting tests on healthy individuals to try to see if it has any side effects. Because the first thing we want to do is we want the drug to be safe, right? The people already have a disease, and we don't want the drug to cause an additional disease in them. So you want the drug to be safe, and so we test in phase 1 clinical trials for side effects on healthy individuals. Then after that is approved, there's no side effects, then we jump on to the next phase. Okay, we get approval for the next phase, phase 2 clinical trials, in which we test on a small number of patients, right? Initially, very small number of patients who are willing to take part in this clinical trial, to test them, test on a small number of patients, check for effectiveness. Okay, see if the drug actually works in the people or not as well. Not only does it work in the cells, tissues, mice in a laboratory setting, but does it actually work on people or not? And then if it does work, then you jump onto a larger subset of people, right? Recruit a large proportion of people, phase 3 clinical trials. And if this succeeds and there's no known side effects, then the result, the drug will get an official approval from the authorities. For example, in the US, it's a Federal Drug Administration Authority that gives approval. Okay? The results of all approved research studies are published in journals, right? Including clinical trial researches, these are published in journals. If a new drug clears all trials and passes the legal requirements, then it is given a license to be used by doctors. Okay? Now, there were a lot of drugs that we used to use back in the day, but now their licenses have been revoked because they have been discovered to cause a lot of either birth defects or some other health concerns. Okay, I can give you some examples of them if you want to Google them. There's something called diethylstilbestrol, right? There's something called chloramphenicol, not allowed to be used in the U.S., and so on. So just to review this again very quickly, right? We have drug discovery, then we have preclinical testing, then we have the phases. So again, number 1, we have the drug discovery. After discovering the drug, we test it in a lab, in a test tube, on cells, on mice, etc. Right? Lab testing. Also called preclinical, before we march on to the clinical testing on people. And after that, lab testing, So after that, we will have the phase 1. This is on healthy individuals. And what we're trying to do really is to test for side effects. Okay? Side effects. After phase 1, we will have phase 2. Phase 2. And this will be done on patients. To test for effectiveness, efficacy. And then phase 3, large subset of patients. Large subset of patients. Okay, and then you get, if this all works out, then the research gets published, and you get a legal approval to use the drug. Okay? And that's really it. So you need to remember all these steps, and you need to know all the trials. And I hope this question shows up because it's a big question, right? It's a probably 4 to 5 mark question, and that would be, full marks for you guys. So review this a couple of times. Maybe you can rewatch the section of the video on this, and hopefully you guys can make me proud and get full marks on this one. So here's a meme to finish things off. When your patient asks how his placebo will help him. If I tell you what happens, it won't happen. Because they're thinking that it's a real medicine, and then some things, they try to tell you that, oh, I'm getting some changes, I'm getting some improvement, even though they're on the placebo. So yeah, that's the meme. And okay, so the next topic is monoclonal antibodies. Okay, so what are monoclonal antibodies? Remember, antibodies are Y-shaped proteins that are complementary to an antigen, right? So we can actually make in a lab or in the industry some antibodies that bind to specific antigens of choice. Some antigens that we want to target, we can make antibodies for them. right? And those are called monoclonal antibodies. So we can make antibodies to target, for example, some hormones to test for hormone levels. We can make antibodies to target some pathogens to check if the pathogen is present or not. We can make monoclonal antibodies against cancer cells and so on, right? So we'll do some examples in a bit, but that's essentially what a monoclonal antibody is. It is an artificially produced, right, purposefully produced antibody that we can use for testing blood or saliva or other samples for antigens. We can also use it for treatment of certain diseases like cancer, and tracers for detecting cancer or blood clots. And we'll understand what a tracer is in a bit. So how can we make monoclonal antibodies? Antibodies, again, of that are complementary to an antigen of choice. So it's very simple. So we're going to start with a mouse. A mouse is a living organism that has B cells, right? B lymphocytes. So here's what we're going to do. We're going to take the mouse and we're going to inject into the mouse the antigen of choice. So we're going to inject an antigen for which we want to make the antibody into the mouse. So inject antigen into the mouse. Now here's what's going to occur in the mouse. The mouse will have B lymphocytes, right? Remember B lymphocytes? The mouse will have B cells. B cells are going to go ahead and divide into B cells. By B cells, I mean B lymphocytes. Are going to divide into plasma cells. So we get those plasma cells that are making those antibodies of choice, right? But that's just one plasma cell, or only a few plasma cells that are making only a few antibodies and so on. But what we do is we take this plasma cell. So here's the plasma cell, right? And we mix it, we combine it with a tumor cell. Remember tumors from a while ago? So tumors or tumor cells are able to uncontrollably divide, right? They have this ability to divide in an uncontrolled manner, uncontrolled cell division. So what will occur here is that you have this tumor cell and you have the plasma cell. You combine them together. This tumor cell, by the way, board examiners like you to know, is that it comes from myeloma cells, okay? Myeloma is a cancer cell. So myeloma cells are cancer cells of plasma cell origin, and we combine it with a plasma cell. Mix them together and we get something called a hybridoma cell, okay? Hybridoma cell. So we want to remember this. The end result we get is a hybridoma cell. So hybridoma cell has two qualities. Number one, this particular hybridoma cell is dividing like crazy, right? So it's dividing like crazy and producing many copies of it. And number two, it is producing all of these antibodies, right? Each of those copies is producing the same antibody that we want. And that's how it works. So again, we inject the mouse with the antigen of choice. Once the mouse undergoes an immune response, its B lymphocytes will produce plasma cells. We extract those plasma cells and mix them with tumor cells. So tumor cells will divide, and plasma cells will produce antibodies, and together as a unit, which is called a hybridoma cell. Let's write that down here. So this is a hybridoma cell. And you need to know all these names, okay? Hybridoma cell, myeloma cells, okay? Hybridoma cells will then produce these antibodies, and we extract these antibodies for use. So how can we use these antibodies for specific situations, right? That's up next. So very quickly to review how we make these antibodies, an antigen of choice is introduced into a mouse. The B lymphocytes of the mouse differentiate into plasma cells, okay, and then start making those antibodies against the antigen. Now plasma cells are extracted and merged with a myeloma tumor cell, and you need to remember this, okay? A myeloma tumor cell, myeloma is a cancer of plasma cells, so that these are tumor cells of the plasma cell origin. To make a hybridoma cell, and you need to remember this term as well, hybridoma cell. So hybridoma cell is a hybrid of the plasma cell merged with the tumor cell. So you have both the qualities. It is making antibodies and is rapidly dividing. So hybridoma cells will then be cultured, given the nutrients and everything, and antibodies would then be extracted from them for use. So uses, one of them is we can use them for pregnancy test kits, okay? Pregnancy test kits. So how will that work? Well, pregnancy test kits contain monoclonal antibodies that are attached to a dye. Okay, so here's a monoclonal antibody, and then it's bound to a, let's say, a blue color dye. Okay, it's bound to a blue color dye, and then you have a lot of these. Now here's what will occur. If you expose this to human chorionic gonadotrophin, which is HCG, this is a hormone that is produced by the placenta, by the fetus, and it comes out in urine, and this can be detected in urine samples of pregnant ladies. So HCG comes and binds to monoclonal antibodies on the pregnancy test kits. What this will do is that it will cause accumulation of all the antibodies at a particular place, which will form a blue-colored line on the pregnancy test kit. Okay, so let's have a look here again. So this is a pregnancy test kit, a home-use pregnancy test kit, and the pregnant individual will go ahead and pee on this, and the pee will contain HCG, right, which is going to go in. And then once HCG goes in, it will cause this clumping situation, this fixation situation, forming a blue-colored line on the pregnancy test kits, which means that the test is positive. It does contain HCG, and there's a good likelihood of pregnancy. Of course, then you'll have to do further testing to confirm this, but yeah, this is the initial test that one does. Another example is pathogen detecting. So pathogen detecting, monoclonal antibodies against pathogen antigens can also be used. A very popular example of this is hepatitis surface antigen and hepatitis virus infection. So somebody, let's say he or she has gotten hepatitis virus, one thing that you can do is you can test their blood for an antigen that's present or not. So for example, if this antigen is present, you have monoclonal antibodies binding to this antigen, it will appear on the test kit, right, just like a pregnancy test kit, and give you a positive test result. Okay. Number 3, they can be used for cancer diagnosis. How? Well, for this, you have to make something called a tracer out of the monoclonal antibody. So remember, we have a monoclonal antibody, and this time we have made the antibody against cancer cells, right? So we use the antigens in the making process for cancer. So these antigens here, we use them for cancer. And now we have monoclonal antibodies against cancer. Now, the next thing we do is we attach a radioactive material, right, a radioactive probe, a very small one, onto this antibody. Okay, so we have many of these, and all of them would have this radioactive probe attached to them. Now, the purpose of this is that this will now start producing radiation. Okay, so it's producing radiation, and this radiation has a small half-life, so that you can use it in a human being without too much DNA damage. So you have this special kind of antibodies that bind to cancer cells and are producing radiation. Let's say somebody comes to your clinic and they have cancer, or they are susceptible, or you think that they might have cancer. So one thing that you can do is you can send this, test them using these radioactively labeled antibodies. And what you will do is, for example, this is the person, right? And you think that they have breast cancer. And I'm just going to draw a tumor here, right? Breast cancer. Now, what will happen is that once you inject in this person, the monoclonal antibodies that are complementary to breast cancer, they're going to go everywhere in the person, right, everywhere throughout the blood, but they will accumulate especially where the tumor is, right? They're going to go everywhere, but they will accumulate where the cancer is. And once they accumulate, you can now put this person under a scanner or a detector that is able to detect where the radiation is coming from. Okay, so now this person is here, and the radiation is coming out from this place. So this is detected by a computer, and you get an image. And you get an image, and that tells you that the cancer is at this place, right? Right here on the right side, maybe inside the lung, or maybe inside the breast, and so on. So that is a tracer. A tracer is a radioactively labeled antibody probe that is able to bind to complementary materials and can be used in different things like cancer diagnosis. You can also make antibodies against blood clots. So you can make monoclonal antibodies that are complementary to blood clots, and do the same thing with them, right? You attach them with a radioactive material, making radioactively labeled monoclonal antibodies that are complementary to blood clots. Let's say a person has a blood clot over here. Then these antibodies are going to come in and bind right next to the blood clot and produce radiation there, and that helps you detect that, okay, there's a blood clot here, because the blood clot monoclonal antibody tracers are binding and accumulating here and showing an image at this place. Okay, a lot of radiation is being emitted from this place. So that is how we use monoclonal antibodies tracers in cancer diagnosis. Here's another very fun thing. You can attach, instead of attaching, okay, you can use the radiation and directly target radiation in this way. But another thing you can do is you can attach drugs to these antibodies, very small drugs, and you can attach it to them and then send it to the cancer. And the cancer will, and the drug will be able to better target cancer as compared to other cells, reducing the amount of side effects and so on. Okay, so tracer monoclonal antibodies against cancer antigens essentially are joined with a radioactive probe, which is generating this radiation that is being emitted and detected. So these tracers are injected into the patient, and the tracer monoclonal antibodies accumulate at the site of cancer. Once they accumulate, then the person is placed under a detector to screen for the location of radiation emission. Wherever the radiation is being emitted, that's the site of the cancer. Okay, so it will reveal the location of the cancer, and same thing you can do for a blood clot, for example. So here's an example, right? This is an example in which you get this emission at this place here. Here's another example of a person who has metastasis of a cancer to different parts of the body over some months. So maybe this is at presentation, right? And then maybe this is 3 months later, and maybe this is 1 year later. Okay, so in this case, in the first one, you have only a little bit of metastasis into some bones, and then it keeps on spreading. The cancer is spreading, and this person has, so many places where the antibodies have accumulated and produced this radiation, giving you this image. Okay, this is called a PET scan, by the way, just in case you want to research on this. Not in your course all that much, except for the name. Locating blood clots. Okay, so similar radioactively labeled antibodies can be used to locate blood clots. Instead of locating cancers, you just make antibodies that are complementary to blood clots and send them in the individual. So this will bind and accumulate at the location of the clot, which will be revealed by scanning for radiation emission. Okay? Very, very simple. All this is very simple. So I hope you guys can review this a couple of times to just help store this into memory before you go into the exam practice and the questions practice. And number 5, cancer treatment. Anti-cancer drugs can also be attached to the antibodies, enhancing drug delivery directly to the cancer cells. Right? Remember, chemotherapy, right? Radiation therapy, all of those are very toxic to other normal cells, which damage other normal cells and cause a lot of side effects like alopecia, which is hair loss, and nausea and vomiting. But if you use anti-cancer drugs in this way, then they can be better targeted directly to the cancer cells, avoiding as many normal cells as you can, and therefore lesser side effects. Okay? And yeah, so I think this pretty much concludes our session on the disease prevention and treatment. And I encourage all of you to review this a couple of times, review all the videos a couple of times at least. Make your notes, or at least annotate on your purchase notes. And yeah, I'll see you guys in the next one, okay? Take care.